All right, hi again. This week we're going to just have a very short lecture here just to finish off the story of infectious agents. I want to introduce you to two terms to start off: pathogenicity and virulence. The distinction between these two terms is subtle. Virulence is a relative measure, so we can compare organisms on the basis of virulence. Virulence is the relative ability of a pathogen to infect a host and cause the disease. This is a common misconception, but virulence is not a measure of the severity of the disease. So, for example, rabbit fever right here, which isn't actually a terribly severe disease, so it's a mild disease, is actually the most virulent. It's very easy to cause a disease and infect the host. This is really important. The degree of virulence is determined by virulence factors. Virulence factors are traits of the pathogen that interact with the host and determines how, 1, it enters the host, 2, how it adheres to the host cells, 3, how it gains access to nutrients from the host, 4, how it's able to avoid detection and fighting the host immune system, and 5th, how it actually is able to prevent removal from the host by the host's immune system. In contrast, pathogenicity is a Boolean. It's a yes or no thing. Does the organism cause disease? If it does, yes, it's a pathogen. If it doesn't, no, it's not a pathogen. Virulence is how easily it can be pathogenic. I want to give just a few examples of virulence factors to just show you how these work. So here's some enzymatic examples of virulence factors. In these cases, these are secreted proteins that help the pathogen dissolve chemicals or body structures. The first example I want to describe is called hyaluronidase. And in this case, we're looking at an intact epithelium here. And if you recall, there are junctions that are holding these cells together very tightly. And they're also holding the cells very tightly to the underlying basement membrane. The enzyme hyaluronidase, which is produced by bacteria, will digest the junctions between the cells, which will open up a space that will allow the bacteria to get down to the basement membrane. At the basement membrane, which you recall is mostly made up of collagen, the bacteria will start producing collagenase, and collagenase will then digest the basement membrane, which will give the bacteria access into the underlying connective tissues, which is where they want, which is where all the good nutritious stuff happens to be. So hyaluronidase and collagenase are two examples of virulence factors that help the bacteria gain access to the inside of the body. Another example of a virulence factor enzyme is called coagulase. Coagulase in orange here will cause a blood clot, cause your body to form a blood clot around the bacteria. Now what this does is it hides the bacteria. Your immune system is not going to be able to find the bacteria because the bacteria is held within sort of what your body thinks is a naturally formed blood clot. After your body has stopped looking for the bacteria will then release this enzyme called kinase, which will break up the clot, and now the bacteria is free to enter into your bloodstream and initiate an infection or gain access to whatever damaged tissues that are nearby. To weaken the host, and bacteria and other infectious agents can also release toxins. A toxemia is when toxins enter the bloodstream and affect many sites in the body. So, in fact, sometimes the toxemia can infect parts of the body that are quite a long ways away from the site of infection. So a toxin, by definition, is a chemical that harms tissues or triggers host immune responses and cause damage to the host. Toxins can be brought into two groups. In bacteria, there's an exotoxin, and then there's an endotoxin. Exotoxins destroy host cells or interfere with host cell metabolism. And a good example of this is a cytotoxin. So you see here the bacterial cells, as they're alive, are secreting these exotoxins in red, and the exotoxins in red end up destroying some of the cells that the bacteria is trying to get past in the host. So that's an exotoxin. It's produced by a living bacteria as part of its normal metabolism, and it's designed to do specific harm to the host cell. Endotoxins are a little different. Endotoxins are found specifically in gram-negative bacteria, whereas exotoxins are more common in gram-positive bacteria. And in the cell wall of the gram-negative bacteria is the location of the endotoxin. In particular, endotoxin is created by something called lipid A, which is sort of the active component of the endotoxin. The idea here is that the bacteria is not actively releasing endotoxin, but if you were to kill the bacteria, like say through phagocytosis, that would release the endotoxin, and the endotoxin then gets out into the body, will travel through your bloodstream, cause a toxemia, and cause a huge amount of problems, including fever, inflammation, diarrhea, shock, and abnormal blood coagulation. So a huge point of distinction, again, exotoxins are produced from gram-positive bacteria, they're produced from living cells, and their point is to destroy the target cell. Endotoxins are naturally built into gram-negative bacteria. They are only released when the gram-negative bacteria is killed, and their job is to, again, just like the exotoxin, is to destroy and weaken the host and make it more accessible for other bacteria of the same species. This also creates a problem. So, for example, if a patient has a gram-negative infection, E. coli, for example, is a gram-negative. Let's say the patient has a very severe gram-negative E. coli infection. Do you give that patient a whole bunch of antibiotics to kill that E. coli? We've already talked about one reason why you might not, is that you don't want to kill off the intestinal bacteria to allow for, say, a C. diff infection to take over. But here's another example of why you don't want to overload the patient with antibiotics. If you overload the patient with antibiotics, you'll kill all those gram-negative bacteria, which means you're going to dump a whole bunch of endotoxin into your blood. And in fact, the endotoxin, the toxemia that results from that, could be much more severe than if you just let the infection carry on its course. So again, you have to have a very serious conversation and consider the situation very carefully before you decide to administer antibiotics. We also have a third reason not to administer antibiotics, of course, which is that resistance story that we talked about in the previous lecture. The last virulence factor I want to talk about are called antiphagocytic factors. This can happen in two ways. Remember, in fact, I would suggest if you don't remember the stages of phagocytosis, you pause this and go back to the previous video and review the stages of phagocytosis. So you can block two stages of phagocytosis. First, you can actually just block the attachment of the bacterium with the phagocyte. So the bacterium, which is here, has a special protein layer surrounding it called a capsule. And the capsule makes it not able to bind to phagocytes. So instead of the cell getting phagocytosed, instead it just leaves, and the macrophage is unable to do its job. The second possibility is the cell can survive phagocytosis, and so it doesn't fuse with the lysosome, and instead survives inside the phagosome. So they can get inside the phagosome, and now they're totally protected from your immune system because they're inside a cell of your immune system, and they can just reproduce and reproduce and reproduce and reproduce until eventually they just burst this macrophage, and then they can spread out. So this is referred to as incomplete phagocytosis. All right. So with that's our review of infectious agents. I do have one more lecture that I'm going to post a little bit later in the week where I just review the blood cells and the various processes of hematopoiesis. And I'll see you then. Take care.